I just feel like genetics is so fun. It's one of my favorite parts about biology. Do I say that about every part? But it's also some of the toughest stuff. Just because it's so detail-oriented processes. You know what I mean? I was grading your Punnett squares this morning. They were really good. Really, really good. So all of you that were doing those Punnett squares and turned them in, I just was getting them graded. I know, blame testing, but they looked stinking awesome. You guys are doing a really, really good job. So that made me feel good about what you were doing. Yes, I did get it. It looks great. Perfect, Pip. I was actually really impressed by what you were doing is you're putting not just homozygous or heterozygous. If it was homozygous, you were putting homozygous dominant or homozygous recessive. Nope, Riker, it wasn't. It was not required. It's extra credit. But I still wanted to get them graded in the grade book.
Yeah, I haven't checked yet, but I might check if I got my grade back for it.
You should have, because I did it this morning. You got a perfect score, so don't worry about that. So today we're going to look at something that has to do with DNA and RNA. All right, DNA. Can somebody tell me what DNA is? What is our DNA? Brain refresh. Brain detox, retox. Yes. So DNA is double-stranded. It's a double helix. All right, so we think of it like two strands, like a ladder, twisted. A twisted ladder. That's what our DNA looks like. What is our DNA for? Yes. What kind of information? Yeah, they're the building blocks of us. I'll check into the grade book and see what's going on. Yeah, they give us all of our Yep, they're all of our genes. They tell our cells how to build us, right? Our DNA is like the instruction book for how to make you. All right, just like a recipe or an instruction manual. You're so welcome. Right? So we have our DNA. Who remembers what RNA is? It's the same but a little different. Anybody remember what RNA is? Ooh, yes. Whoop, whoop. This is making me happy. Single stranded. All right. Even if that's the only thing you remember, I'm thrilled that you remember DNA is single stranded. Yes, it's used to convert for proteins. And that's what we're going to talk about today. Perfect, Faith. Oh, I love that. DNA couldn't get the genetic message to our cells without RNA. Right. All right, somebody read our goals for us today. Somebody want to pop on the mic.
Oh, yeah. Oh, my mic cut off. Oof. I hate it when Neuro does this. Anyways, if no one else wants to, I shall do it. So, what is transcription? What is the product of transcription? And where does transcription occur?
Perfect. So we see again all these guys down here. We're going to start over today with learning new stuff. So we're starting at the bottom of this triangle. And we're going to build our way up this week, okay? So we're going to start at what is it at the base of that triangle. And then by the end of the week, we should be able to start applying it, analyzing it. All right, we shouldn't be all the way up here. Most of us probably won't be up here or even up here. We're going to try to get up here into orange and yellow by the end of the week. All right, let's take a peek. This is a good slide that's going to help us review. Yes, so our DNA, it is double-stranded. All right, it's talking about bases. What bases is it talking about? It's talking about nitrogenous bases. Nitrogenous bases, what are they? It's kind of a fancy word for a bunch of what? Yes, Arya. Yes, for a bunch of letters. So we have adenine that pairs with what does it pair with in DNA? Good. Adenine pairs with thymine and cytosine pairs with kind of looks like a C. Yes. Guanine. Group effort. All right. So in DNA, we have those are our nitrogenous bases, right? Our adenine pairs with our thymine. Our cytosine pairs with our guanine. And then there's a sugar called deoxyribose. All right. Deoxyribose. RNA is a little different, right? One, it's single stranded. And then instead of this thymine, it has uracil. So in RNA, instead of A pairing with T, you'll never see A-T. If you see A-T, that's an indicator that it's DNA. All right. If you see adenine and uracil paired up, that means that it is an RNA molecule. Mommy. Right? That's like kind of a quick way of knowing. Yes, Ren. Look what you made me do on my Oreo. Oh, I made you put Oreo on the couch? Yeah. That's weird because I'm not even over there.
Oreo? Is that Raja's brother?
Oreo is actually an Oreo cookie that she's eating for breakfast. This is what happens when I am teaching in the background.
That's the thing.
They do all kinds of ornery things behind the scenes. You may
The joys
not
of working
come
at home
here this morning,
and
Ren,
being
because you are not properly dressed. So if you get yourself properly dressed, you may say hello. You are not properly dressed this morning. They ate yogurt and a banana for breakfast, but they raided their Christmas cookies. We had a giant Christmas baking party this weekend, and we made icing cookies and gingerbread cookies and Oreos dipped in chocolate and Nutter Butters dipped in chocolate, lots of sprinkles. What else did we make, Mila? You want to show them your cookie, Mila? Mila's about to eat one too. You want to show them your cookie? Mila is properly dressed, in pajamas, properly dressed, but you may have one cookie. Okay?
Okay.
And Ren, no more cookies, and no more cookies on the white couch.
Yes, Mom.
Oh, they love them, Milo. They love your cookie.
Thank you. I asked it and split myself.
She said, I iced it and sprinkled it myself. Yes, it was so much fun. Ren, get away from that cookie platter. I know. White couch. Why did I buy a white couch? Not smart, was it, Riker?
You had a yellow couch. That was much better.
We had a beige couch. It was not an intelligent decision. I had this great idea that I would put some Scotch Guard on it, and I was like, they're old enough, right? Like that I can say, don't eat Oreo cookies, chocolate dipped Oreo cookies on my white couch. But then I forget that they're doing all kinds of crazy things. You may not come on the camera, but you want me to show your cookie? Here's Ren's cookie. She wants to show you one too. She's jealous of the praise. This is a gingerbread cookie, a nice gingerbread cookie.
Mine was a snowman.
She has some gold sprinkles on there.
And that's what I call creativity.
Woo! I just broke his leg off. Oh no, you better eat it.
I don't want to eat it.
Oh, then I'll eat
I
it.
guess we have.
We must sacrifice his leg. Don't
We
come
have
to my
caught the gingerbread man, but had to sacrifice his leg.
house. Don't come to my house. If you break your leg, we might just eat it. All right, guys.
When you said a person's leg fall off, the other person's leg.
Come to our house. Your legs are falling off, and we're eating them. It's not a good situation. No. Awesome. So I know. I'm sorry. Distracted. They missed you too. You missed up my class too, didn't you, girls?
Yeah.
Yes. They missed you. So, yes. So we are back to our RNA. DNA and RNA single stranded versus one. DNA is double stranded, RNA is single stranded. The biggest difference then is in our bases. Our bases, we're having adenine and thymine versus adenine in uracil. All right. So, what are we looking at today? We're looking at why do we even have RNA? Yes, no, we're leaving no evidence of that. Ooh, a chicken leg. That's true. You can eat chicken legs too. Mila does not like chicken legs. She said does not approve. So what RNA is, I want you to think about this. RNA it equals a copy of DNA. Everybody type that. RNA equals a copy of DNA. Okay. So what happens is, do you guys have documents at your house that your parents like keep in a safe spot? We have like a fireproof safe that we store somewhere secretly in our house, and it has like the copies of the purchases of our cars, the like information for our house when we signed all the information to buy our house. It has our living wills in it, so like anything happens to us, it tells what happens to our things. Yes, your parents' room. It is in my room. It is a safe. Yes. Yeah. What about technology? What part of technology? There are some safe stored documents in our technology.
Oh yeah.
That's
I
perfect.
mean, I want to tell you guys a quick story.
Sure.
So basically, what I was saying about I had some iPhone 4Ss when I was young. I have two of them stored secretly, but and apparently, one of them out of the three that I had in a bag works perfectly. It was broken because I smack dabbed it, like threw it on some soup can. It broke somehow, but it still works and it works perfectly fine. Anyways, so as I was looking through it, I had found it's not documents, but I don't know if this counts as something that you put
Yeah.
in a safe place. But while I was looking at it, there was stuff, a lot of stuff on it. Photos. There was one video, but it was short, and I found some things. I was like, okay, you know what? Let me take a few pictures, and then I showed the proof. I have a few old things from years ago, and this is one of them. This is Angel. I have a few.
Oh,
I used to have this app all the time when I was
that's
young,
awesome.
and I had that's one of them. This is Jason De Leon. He is a cool guy, He's in his wardrobe room, just chilling. And I also have this one. I somehow kept them all over the years.
That's so cool.
And then there's this one, Nini. He's just any other guy, really. So yeah, I have kept so many old things over those years. I wish I could go back and do that all over again. So yeah, that is just
That's so cool. No, and that's a really good point. We can't store things like on USB sticks or on Google Drives, things that we don't want to lose. I say like backup pictures in safe spots. That's perfect, a perfect example. So things that are important to us or things that we want to keep, we put in a safe spot. And that's what our cells do with our DNA. It goes in our nucleus and it has a membrane around it, and our DNA stays in the nucleus. Our DNA does not come out. That's the safe spot, because if we lose the DNA of our cell, we lose the information on how to operate, and it's death, right? We don't have it. So what RNA does is it goes to the nucleus, knocks on the door, right? Or it knows the key to get in, and it goes in and it makes a copy of the section it needs. Because if it made a copy of the whole thing, that would be so much information that it doesn't need. It just goes in, it finds the spot it needs, just kind of like a photo album. Like sometimes I'll go back in my digital photo album on my girls' birthdays and like pull their baby pictures, right? Anybody have parents that will do that? So I'll look at their baby pictures, or like a cookbook, right? I keep all my recipes in the cookbook to keep them safe in a place where I know they're at, because I don't want to lose them. They're valuable. They're the recipes that my family likes, that have all the instructions to everything we love. So we go there. If you want to, go through your things tomorrow. All right, go through some of your photo books or your stored digital things, and bring a baby picture with you to class tomorrow. Definitely not mandatory, just fun, only if you want to. You can send them to me. You know what we could do? Pip, do you have a spot on the do you think you could put a spot on the Padlet? She might have stepped away for a minute because I'm pretty sure she'll be all over that. All right, maybe we'll put a spot on the Padlet, and you could bring a picture, a baby picture with you tomorrow. Because that represents what? Going back to a safe place and being able to get a copy of something. All right, so what does our RNA do? It goes in and gets a copy of that DNA to help go make what we need for our cells to survive. It does. It does. So that is what the exact process that we're going to look at today. Let me just skip this. It's a temporary copy. So what happens is it's going to go in and make a temporary copy of the DNA. So here we go. The goal when we make proteins, the first thing is we have to make our mRNA, our messenger RNA. So our messenger RNA, you can see right here. Transcription is the first process in making a protein. All right. So right here, transcription is the first step in making a protein. And during transcription, it has nothing to do with the protein. It has everything to do, girls, you need to use quiet voices. Has everything to do.
Yeah, we're in class right in Iowa.
Keep it down. Keep it down. It has everything to do with making a copy of RNA. That's what transcription is. Making a copy of the DNA to RNA. And it happens in the where? What is this spot in the cell? What are we looking at right here? Yes. Perfect. This is the nucleus. All right. The DNA is in the nucleus. The first step in making a protein is transcription, and that is to get the code from DNA. So the mRNA comes in and starts at a spot in the DNA and will copy it into a certain point, and then it snips and takes that and leaves. Yes, Pip, if you would make a spot in our Padlet for baby pictures, we're going to go back to our, just like our, in making a protein, the first step is to go back to the records, right? And get a copy of it. I would love if anyone wants to do this, go back to their records, right? That their parents have kept, their baby pictures, and get a copy of that baby picture. Take a snip and either put it in our Padlet or send it to my email. And I knew you would be all about our community biology community fun coordinator would put that in the Padlet for us. All right. So what happens when transcription inside the nucleus occurs is there is an enzyme is a protein, all right, and it unwinds it. See this right here, how it kind of looks like teeth? It unwinds a section of the DNA. And it says, there it is. There's a copy for making that. That's the recipe I need right there. It unwinds it. All right, so this gold strand right here is the DNA. See how that is? This is the DNA. The RNA forms based on the DNA. Look what it does. It's just like matching. If there's an adenine, the mRNA adds a uracil. And then it says, okay, there's a thymine. So I need an adenine because, right, RNA does not have any thymine in it. A and U paired together. So anytime there's a T, there will be an A. Anytime there's an A, instead of being a T, there's going to be a U. Good. So that's all transcription is. It's matching. It matches up these base pairs. So if we know that G and C go together, we can do transcription. If we know that A, T, and U go together, we can do transcription. So it goes until it gets to the end point where it's done being unwound. All right. And then there's actually like a enzyme that tells it to stop. All right. And then once it's done matching up those base pairs from this DNA copy, this green RNA strand will leave the nucleus. That's all we're doing today is transcription. Transcription. Transcription happens in the where of the cell? Where does that happen? You got this. Yes. Everyone say nucleus. It happens in the nucleus of the cell. All right. First step is to unwind the DNA. And the second is to match up those base pairs. All right. We unwind and then we match up. Unwind and then we match up. All right. We're going to do it. All right. I'm a doer. Does anybody else learn by doing things? I kind of have to do things. All right, so let's make some lines here. Don't start tatting up my board yet. I want you to do it though. Just give me 1 second. I want to. All right, so this is the section of DNA. I want to make some lines here. I want some of you to help me. And we're going to do what is the RNA? If this is the DNA strand that was unwound, we are going to be the transcribers. We're going to be the mRNA today, that messenger RNA. So let's do this first one together. If the DNA reads A, here's our RNA strand, our mRNA. If the DNA reads A, what's going to match up? Good. So in DNA, it would absolutely be T, but we have to remember that we can't. Let's write that up here because that's the trickiest thing. We have to remind ourselves that there's no T in uracil or in RNA. I would actually, if I was taking a state test, I might actually write this down on a piece of paper while I'm doing a question because it's going to remind me that. Because sometimes I need a visual reminder as I'm doing things. What is it going to be instead of a T? It's going to be a U. So that anytime I go to accidentally write a T, that I write a U. So good. So usually we match A with T, but because it is RNA, our very first nitrogenous base is a U. Good. It's a U. All right, what would be next? So we're going to go back to our DNA. Our DNA says C. You guys are awesome. So this would be a G. Perfect. Transcribers. G. It's kind of like coding, right? Is C. This is exactly what happens all the time in our cells. When our T. Good. And this is the tricky one. Everyone's like, oh no, it's that thymine. There's not thymine in RNA, but what pairs with thymine is adenine, and there is adenine in RNA. So that's the tricky part. The A is there. That's perfect. It didn't trip most of you up. We still have our adenine, our cytosine. Perfect. Matches up with guanine. And lastly, good. It's our adenine molecule. Adenine usually pairs with T, but it's RNA. So we're going to match it up with, what do we replace the T with in RNA? Good. U. Perfect. U. Awesome. That is transcription, guys. You're doing it. That should make you feel really good. All right. Can somebody lead us through this one? This is another example. Could someone lead us through?
Me and Aria have our hands up.
Do you guys want to do it together? You want to make a halfway mark?
Yep. All
Let's
right.
do half.
There.
Is
Let's see
halfway.
if there's any even point first. So if I can calculate correctly.
Is that even?
Oh, I think it's an odd number.
Here, how about we just do it where that line is?
Yeah. Oh, great job, Zach.
Isn't it?
Zach knows.
He's going
Anyway,
for it.
Oh yeah, I need tools alongside Aria for this.
Oh, I'm so sorry.
No, you're fine. You're fine. All right.
What?
All right. Sounds good, Ari. Aria. I'm just gonna call you Aria because it sounds feminine in my opinion. Anyways, I'll take left. Aria will take right.
Sounds good. You're up.
Now I have tools.
Perfect. All right, go ahead and lead us through. All right. So they're working through there. Perfect. Now, will you ladies, will you go through and explain, just read through and how you did it and what you did?
All right. I'll read the left side. Aria will read the right. So this is mine. Looks pretty beautiful. Like so organized, really. And so AU, you know, what was that? I know uracil binds
Yes.
with the A, which I forgot what the A was. Like something starts
It's
with
all right. It's
A.
adenine, but that's fine. You don't need that.
Adenine. Okay. Adenine pairs with uracil, and the T pairs with adenine. I forgot what the T was. The G, which is I remember the name, but I know C is
Guanine.
guanine. Okay. Guanine pairs with cytosine. Cytosine pairs with guanine. Adenine pairs with uracil. The T pairs with adenine again, and then guanine pairs with cytosine. And Aria shall do the right side for explanation.
Awesome. Thank you.
All right, so okay, let me use my tools really quick. If it works, there we go. Okay, so for C, I know C pairs up with G, so this is obviously doubled right here. So if one makes C, then other makes G. I just doubled it. And then A pairs up with U because this is RNA and not DNA. And then, like you said earlier, if there's a T, T matches up with A, but because RNA doesn't have a T, then we only focus on the A part. And then G and C pair up again, and C and G pair up again. And then there's A and U, and T and A, and the same thing with this T The T doesn't matter in RNA, but the A does.
I am so proud of you. Good job. Please, guys, give them all kinds of praise and love. It's Monday, and that's a lot of brain work. And you guys just did transcription. Transcription is what you guys just did. You mastered transcription. I would go to say that you already are moving up that triangle of understanding. One thing I like to do as a teacher when I check back like my students' work, a quick check for me is always if you're doing a DNA.
Muted!
Yeah, I had to yell at my kids. Sorry. I said girls. Right here is I look really quick at this line and I don't see any T's. So that is a really good sign to me, right? Because if I'm looking for you guys to transfer to RNA, I know that there's no thymine. So a quick check for your understanding is for me to eyeball that uracil line and it's like perfect. They don't have any T's. That's a good sign. I know I have to remember that you can still see me, all right? I can yell, but the mean mom face still shows. Oh, gotta love it. Sorry, just real life. It's a Monday here too, guys. And you know what? We talked about adjusting pre-class, and that I think that is such a just real life thing to realize. Like, life is hard, and we have to pivot a lot, right? And with testing and school, it's been a lot, and sickness here, it's been a lot of pivoting here too. Sometimes it's hard to adjust to schedule changes. So please forgive us today as as the written home adjust to scheduled changes of live lessons again. We appreciate you. All right, one last thing, okay? You know why these quick checks are good. Why are these quick checks good? Where do you usually see these suckers again?
You never know if they could be on a quiz or not. You could even also be on our, exam soon.
Absolutely. So it's a good thing to screenshot because a lot of these times, and I mean by all of the times, these same questions are your quiz questions and your test questions. 100%. I don't write these questions. I take them from your quizzes and tests. So because sometimes they're just worded weird. Agreed? They're weird questions. So this helps us preview those. So, anyone want to read one of these questions? You can hand out some questions, question reading today.
All right. If no one else wants to, I will read one. So
Go for it. Let's read this first one.
Okay. Which action is one role of RNA polymerase during transcription? A, to break down an already existing protein. B, to produce a start sequence. C, to build a molecule of mRNA. Or D, to switch uracil with thymine, which I finally know what the T stands for, on an exposed gene. Thank you. We shall use
Perfect.
pointers for this one.
Yes. So it says which action is one role of RNA polymerase during transcription? Oh, I'm going to sneeze. I'm sorry. Sorry, I felt it coming. So let me see if we have a slide that we can come back up here to. Oh, is it this one? Hold on one second. We're going to go back up. So all RNA bases bond polymerase to expose pieces of DNA. All right, so RNA bases bond polymerase to expose those pieces. So they're kind of like glue. Does that make sense? Polymerase is kind of like an enzyme that's like glue. So if you had a genetic glue stick, it would be polymerase. So polymerase is in charge of like keeping things attached, keeping things together and organized during the process. We didn't really highlight that as we were going through it, so I want to make sure. So is it to break down to produce a star sequence to build a molecule of mRNA or to switch thymine? What do you think? Think about glue. Yes. Yes. Right? Glue is important for building. Yep. So C is the right answer. It is to help us build an mRNA molecule. Polymerase is like the glue. All right, that holds things together. Perfect. All right, what about the second question? I'll give you polling.
Oh, I'll probably read this if no one else wants to read. All right. Which statement is true about the DNA molecule during transcription? A, it opens up all the way, one gene at a time. B, it unwinds from both ends simultaneously. C, it makes a copy of itself. Or D, only a small area is opened. Thank you.
Good. So what happens during DNA? Does the whole DNA unwind? Does it unwind from both ends? It makes a copy of itself, or only a small area is opened? Does the whole DNA unwind? I want you to focus on in this question, it's more of a space question. Because it would be total chaos if we unwound all the DNA. I know, Pip. I think people got used to not having class. And now we have class. They'll probably trickle in through the week. So the answer is actually D. Okay, only a couple people actually answered it correctly. And that's because, I mean, it's an important question, but it's a small part of what we actually talked about today. So let's go back to this picture, because I think this picture helps with the understanding of that. Where's my picture? This one. All right, see how right here only this small section of the DNA is unwound? It's only a small piece. So whatever protein that we are going to make, just like if I open up my family recipe book, I'm only going to go in and look at one recipe. I'm not going to go in and look at the ingredients that I need for the whole cookbook. That would be so overwhelming. Can you imagine how many ingredients that would be if a cookbook gave you all the ingredients at one time? Yeah. And it doesn't matter if we get these all right or all wrong, because this is helping us learn. And like I said, some of these questions aren't my favorite questions, but they're the questions that they're going to ask you. So it's just good to go back and revisit that. Does that make sense, though? Let's do a thumbs up or thumbs down. Or red check, green check, red X. It's only going to open up that small part. It's only going to give a small copy of its information. It's not going to overwhelm the cell with all this information. It's just going to give them exactly what they need for what they're making. It's going to unwind a very small part. And a lot of times these questions are going to ask you, give you answers like this that just like it unwinds from both ends, or it unwinds from one time, or it, like, these honestly are all kind of just trick answers to try to get where we're really just focusing on this. I believe it only unwinds from one point, in case that matters. My girls, keep being sweet. All right, keep your hands to yourself and have nice hands. All right, last question. Let's go through it, and then we will call it a day because we're close on time.
If the part of the DNA gene being transcribed is TGCAAT, what will the mRNA code be for that part? A, TGCAAT, B, UGCAAU, C, move the pointer, if you can, ACGUUA, or D, UCGTTU. Thank you.
Thank you. All right. And this is what I would do. If I was answering this question on any test, I would get a piece of paper and not look at those answers because that is a lot. That's overstimulating to me. So I would go back and do just what we did earlier with the matchup. All right. T pairs with A usually, but this is RNA. So it's U. G
Oh,
pairs up
and
with
Aria
C,
would like to get on mic if you don't
E,
mind.
U, U, A. Go ahead, Aria. Yep. Go for it, girl.
Yeah, so what I was just gonna point out really quick is like process of elimination really helps me. So like with the TU and TA thing, if you know, like if you just remember that part, I feel like it's pretty easy to eliminate the other answers because like obviously A wouldn't be correct and obviously C wouldn't be correct because of the A.
Yes, I love that. So Arya is using what she knows to eliminate what she knows is not right first. That's a super smart strategy, right? To go in and say, I know RNA doesn't have any thymine in it. So if there's any answer choices with thymine, I can get rid of it. And then if you know that if when you're pairing A, T, look, it gets you right off the bat. It says, do you know that there's no uracil or that T pairs with A right at the beginning? Oh, and I messed this up. Trying to switch it out. This is A. This is supposed to be an A. Trying to do it too quick. Okay, so C would be your correct answer. Good job, good job, good job. I'm gonna pop you out of here fast because we're a little bit late, but I feel like we did a good job accomplishing our goal. Transcription. What is it? We are making RNA. Where does it happen? Where does transcription occur? It happens in the nucleus. All right, and what is the product? Well, the product of it. What are we actually making? We are making messenger RNA. Perfect. You guys are rock stars. I missed you so much. So much. I will see you tomorrow morning, not at 8 o'clock. It will say 8:30 to 9:15.
I hope.
I promise. All right. Have a great day, guys. I will open it as soon as I leave, okay? I'll go open it. So, and Aria and Justin, Pip, whoever it is, you guys can let me know if it doesn't open. Oh, I missed you guys. Have a great day. Thank you so much. I'm glad you missed me too. That means you like me a little bit. See you tomorrow.